Hello, my dear Butcho, and welcome back. I know, exams are just around the corner and the tension must be building up. But don't you worry one bit. Your favorite tutor is here to make this chapter on microbial culture super simple for you.
Seriously, figure not, this chapter is one of the easiest, I promise you. It's a real scoring chapter. If you just listen carefully for the next few minutes, you can easily get full marks here. So just relax, take a nice deep breath, and let's get started, shall we?
All right, so are you ready? We're about to dive into this incredible, invisible world of microbes. They're literally all around us, on your phone, in the air, everywhere. But we can't see them. And trust me, what we're about to learn is more amazing than any action movie you've seen.
So first things first, what are we even talking about? Well, we're talking about microbes or microorganisms. Now here's a fancy word for you, ubiquitous. It just means they are, well, everywhere. In the air, in water, in the soil, even inside us. And the science that studies these tiny little giants? It's called microbiology. See? Simple stuff. Now, believe it or not, for a long time, people had this really strange idea. They called it spontaneous generation. They actually thought that life could just, poof, appear out of nowhere. Like frogs from mud or flies from rotting meat. Crazy, right? It was a big mystery until our hero, the great Louis Pasteur, came along to settle the matter once and for all. So how did he do it? Oh, it was brilliant and so simple. He took a flask, but with a special S-shaped neck. He filled it with some nice broth, and then he boiled it. Why? To kill any germs that might already be inside. Now here's the clever part. The S-bend let air come in, but it trapped all the dust and microbes from the air in its curve. And guess what? For days, weeks, the broth stayed perfectly clear. Nothing grew. But then, he broke the neck of the flask. The microbes in the air could finally get in, and boom, the broth turned cloudy. He proved it. Okay, Butcho, look at this diagram very carefully. This is gold for your exam. It's a pukka 3-mark question, I'm telling you. All you need to remember is that the S shape was the trick. It proved that life comes from other life, not from thin air. It's carried on things like dust. This one simple experiment completely changed how we see the world.
And this is the big one. This experiment led to one of the most important ideas in medicine ever, the germ theory of disease. The idea that tiny, invisible germs can make us sick. This wasn't just some small discovery, It's the reason we have clean hospitals, antiseptics, vaccines, everything. It literally changed the world and saved countless lives.
Okay, so Pasteur proved germs cause disease. Great. But that opened up a new question, right? How do you figure out which germ causes which disease? I mean, there are thousands of them. For that, we need to talk about another scientific superstar, Robert Koch. He basically created a detective's rulebook for catching these microbial criminals. He gave us what we call Koch's postulates. Think of it like a 4-step checklist for a detective. Rule number 1, the suspect microbe must be at the scene of every crime, meaning in every sick person. Rule 2, you have to isolate the suspect and grow it by itself in the lab, a pure culture. Rule 3, you take that pure culture and see if it causes the same disease in a healthy person. And finally, Rule 4, you have to be able to find the exact same microbe in that newly sick person. If you can tick all 4 boxes, case closed, you've found your germ. And look here. This diagram shows it perfectly. This is his actual experiment with mice and the anthrax bacteria. See, he took the bacteria from a sick mouse, grew it in the lab, injected it into a healthy mouse, and that mouse got sick with the same disease. It's a perfect example of his 4 rules in action.
All right, so we know how to catch the microbial culprits, but how do we study them in the lab? Well, just like us, they need to eat. So scientists have to become chefs and prepare a special buffet just for bacteria. You see, to get microbes to grow, you have to give them the right kind of food, a perfect meal with all the nutrients they need. And this special food is called a culture medium. It's basically a yummy soup or a jelly that bacteria just love to grow on.
Now, not all buffets are the same, right? There are 2 main types of this microfood. First, you have synthetic media. Think of this like a master chef's recipe, where you know the exact amount of every single chemical ingredient. Super precise. Then you have complex media. This is more like a hearty home-cooked meal, with ingredients like yeast extract. We don't know every single chemical in it, but lots of different microbes can grow on it.
Okay, now things get really clever. Imagine you're throwing a party, but you only want certain friends to come. That's what selective media does. It's like having a bouncer at the door with a very strict guest list. It has ingredients that let only the one type of microbe you want to study grow, and it blocks everyone else. Super useful, right?
And then there's my favorite, differential media. This one is so cool. It's like a special background that makes different types of microbes show their true colors. For example, on a medium called blood agar, some bacteria will actually eat the red blood cells and create a clear circle around themselves. Others can't, so you can tell them apart just by looking. It's like they're wearing different colored shirts. Okay, so we've talked about Pasteur, Koch, and even cooking for bacteria. But you might be thinking, sir, why does all this matter to me? Well, let's connect all this science to our real lives, because the things we can do with it are, well, they're amazing. For starters, medicines. We use what we know about growing microbes to turn bacteria, like E. coli, into tiny little factories. And what do these factories make? They produce life-saving things like insulin for people with diabetes, and so many of the vaccines that keep us safe and healthy. It's incredible. And what about the food you eat every day? Do you love dahi? Of course you do. What about bread, idli, pickles? All of these are made using, you guessed it, microbes. We grow specific bacteria and yeast to ferment these foods and give them that amazing taste. So next time you have a bowl of dahi, remember to thank the little microbes for their hard work. And last but not least, biotechnology. This is a huge one. Microbial culture is the engine that powers modern biotech. You know, when scientists create genetically modified organisms, like a crop that can fight off insects, they often use bacteria as tiny little delivery trucks to carry the new genes into the plant. None of this amazing technology would be possible if we didn't know how to grow these microbes in the lab. All right, everyone, deep breath. We're at the finish line. Let's do a super fast 2-minute revision to make sure all these important points are locked in your brain for the exam. Ready? Focus now. So, if you're making your final revision notes, what are the absolute most important things to remember? The topics that are almost guaranteed to show up on your exam paper. Let's run through the final checklist. Here we go. Number 1, Pasteur's S-flask experiment. What did it do? It disproved spontaneous generation. 2, Koch's 4 postulates. These are the detective's rules for linking a germ to a disease. 3, culture media. That's just the food for microbes. Remember the difference between synthetic, the precise recipe, and complex, the hearty meal. Then selective media, the party bouncer, and differential media, which makes them show their true colors. And finally, the applications, making medicine like insulin, food like dahi, and powering biotechnology. That's it. That's the whole chapter right there. See? What did I tell you? It was easy, right? You've just revised the entire chapter in a matter of minutes. Just go over these key points one more time on your own, and you are going to do fantastically well. You've got this. All the very, very best for your exams. I know you will make me proud.